Okay, hello everyone, and welcome to a new section of Kernel Notes. Okay, so you should have a handout with you for this PowerPoint or this presentation. It already has a title at the top. I pre-titled it. You may want to add the actual page number at the bottom of the page so you know where to attach these to your interactive notebook. So since it came out on 2 pages of printing, you're going to be putting one page, the first page, front and back, that is the front and back, you're going to label it 64A and B. And the other page, you're going to label 65. And it's a one-sided page. There's space for the summary. Because it's a one-sided page, you can put your summary underneath, so you guys can complete these notes. Okay, so these notes have to do with how geneticists use the principles of probability to make Punnett squares. So you're going to be filling these in, and hopefully it'll take you less time and you can listen to more of the presentation. Okay, so probability. We use probability to figure out the likelihood that a particular event will occur or the percentage. We see this used all over the place. Elections, this is our election season, so we see it during elections. People are trying to figure out, okay, so Iowa, what percentage of the population is going to vote for this candidate or that candidate? They try to, use those a lot. We hear about that a lot. The Super Bowl, even before the Super Bowl, everybody had these percentages of the Panthers winning, and people bet money on those. So percentages, we encounter them a lot in our day-to-day life when we listen to any kind of media. In a more personal basis, heads and tails spinning, when you're like, oh, heads, oh, I want tails, and trying to decide things. What are the chances of each of these things occurring? Well, we call basically there's a 50% chance of getting heads, and there's a 50% chance of getting tails when you flip a coin. So it's a half a chance of one, half a chance of the other. Okay, so you have to ask yourself, what is the chance of getting, if you flip it twice, what is the chance of getting two heads, or both times you get heads? Well, this would be basically how we use probability. What are the chances of something happening? So what are the chances of getting two heads after by flipping the coin twice? And that is something that we can use probability with. In this case, it would be one half, 50% times 50%. And if you do it mathematically, you realize that that is actually 1/4 or 1/4, because you have to multiply the two halves together, and so you get a fourth. So it's 1/4 or 25% chance of that happening. This is the principle of probability, and we can use it more to fit science, because we can use this to predict the outcomes of genetic crosses. We can actually, by knowing the numbers, or the likelihood that's by knowing the numbers and what genes people carry, we can then figure out what the percentages of maybe their offspring carrying the same thing. So that's how we use probability in genetics. Okay, so first we have to establish some ground rules. So an allele, just to remind you from the previous presentation, is a different form of a gene. Okay, and when the genes are the same, with the same alleles, you receive one allele, one form of a gene that's the same as you get from the other parent. That is called homozygous, because the prefix homo means the same. Zygous stands for the zygote, which is basically a technical name for a fertilized egg. So, if the a fertilized egg has the same alleles for certain traits, then we call those homozygous. So, for example, the trait of height, the trait is height. If you get a dominant allele from your mom and a dominant allele from your dad, and we're talking pea plants here, the mom pea plant has a dominant allele, and the dad pea plant has a dominant allele, we term that homozygous, because the zygote has two of the same alleles for that one trait. Or if you get the allele for shortness from the daddy plant and the shortness from the mommy plant, you wind up with homozygous recessive. So that would be the little t, little t. Okay. For the trait of shape, you can get the round dominant from dad and round dominant from mom plant, and you get homozygous dominant, round. Or you could get the wrinkled, the little r from mom, and the wrinkled little r from dad, and that would be homozygous recessive, wrinkled. That would be the shape. Color. The big B and big B would mean that that plant received the dominant from the one parent plant and the dominant from the other parent plant for color. And the two little b's would be that one of the parents gave them the recessive little b and the other parent gave them the recessive little b, and so the fertilized egg has homozygous recessive. Or, okay, so that's the way that's seen. Now, this second case is when you get two different alleles and different hetero. So if homo is for the same, hetero we say different. Okay, so hetero is different, and zygous, once again, the zygote, the fertilized egg. So when the fertilized egg or the zygote has one trait that's different from that they got from each of the parents. So let's say the one plant gave passed down the dominant, the height, tall allele, and the other plant passed down the recessive one, you get a big T, little t. So the height is now no longer, they're still tall. You have to realize the dominant's still dominant. So they're still tall, but the other gene they inherited was the recessive form of the gene. We call those heterozygous. The shape here. So if they received one of the genes, one of the alleles from one of the parents is a dominant one, and the other allele from the other parents for shape is a recessive, that combination would give you a heterozygous. It's still round. Color, big B from one parent, or a dominant one from one parent, and a recessive one allele from the other parent, you wind up with a situation where you've got blue, I'm sorry, brown, because the brown is dominant over the recessive blue. So we've got, they still are tall, round, and blue, but it's only because they carry one dominant allele. Okay. So now we're going to go over the difference between genotype and phenotype. So genotype, the type of genes you have, the genes are in the DNA inside of your nucleus. Can you see them? No, but it is your genetic makeup. Examples of how we show them on paper are letters. So big T, big T, big T, little t, or little t, little t. That's how we show genetic makeup. That would be the genotype. The phenotype is the physical characteristics. These you can see. This is what these things look like, what you look like. Phenotype, physical. They depend on what you inherit or your genotype. And examples of how we write that down would be like tall or short or white or purple or wrinkled or and round or whatever. Those are like words we actually use to describe what something looks like. So that would be its phenotype. Um, and finally, the question here is, how can two organisms have the same phenotype but different genotypes? Well, I give you the situation where you get one person with brown eyes and another person with brown eyes, two people with brown eyes. Let's say one family, everybody has brown eyes. Every grandparent, everyone in their family has always had brown eyes, and they have brown eyes. And you get another person here with brown eyes that half of their family has blue and green eyes, and the other half of the family has some brown and some green. Yet they have brown eyes too, and they look the same color brown as the other person. They may have the same brown eyes, but do you think the probability of the one that has some other color genes in their family or alleles in their family would have a different genotype? And if you know the situation, yeah, how can you tell? Because many of these people that have different colors, even though they have blue eyes, they can produce offspring that have light eyes, despite the fact that they marry someone with brown eyes too. So you get two people with brown eyes, they can have kids that don't have brown eyes because they may still be carrying genes that are not showing in their phenotype, but they are in their genotype, so they can pass them off to their offspring. So we've all seen combinations of this. You may be a result of that, or you might wind up having kids like that, and you can tell that the genes that you carry, sometimes they don't show up in you, but they'll show up in your kids. Okay, so you're going to be doing a check here, and I hope you get these right. You make sure you type them into the right area in your answer key. Just remember, homozygous means the same, heterozygous means different. Genotypes, we use letters, and phenotypes we use actual words. So hopefully those are good enough hints. Good luck. Okay, so here's the, we're going to basically make a Punnett square to show the possibilities of different outcomes. Um, the alleles we're looking at for height, the trait of height is the allele for tall, and we're going to use big T for that, and the recessive short, we're going to use a little t for that. And the phenotype, once again, a reminder, it's a physical characteristic, whereas a genotype is a genetic makeup. So when we look at homozygous dominant, that phenotype is tall. For heterozygous, we get the phenotype being tall, and the homozygous recessive condition is short. And that's the way you read this. So we're going to create a Punnett square using this information. So what you get here is a situation where we're going to cross homozygous dominant with a homozygous recessive. How do you know that? Right there, it shows you that there is a big T, and a little t, little t. That means one parent is homozygous dominant and looks tall, and the other one is homozygous recessive and it looks short. So if you have one mommy having big T, and daddy plant having little t, what is the probability of them having offspring? What kind of offspring can they have, tall or short? So we can cross them. And the way you do that is that you basically you bring them together. You multiply big T times little t in each of the boxes, and you wind up, once you do that, you're going to have 100% possible genotypes, 100% are heterozygous tall. The possible phenotypes is if you look, if all of them have a big T, at least one, that's dominant. So 100% of the offspring of this genetic cross would be have the probability of being tall. So that's what this gives you. It gives you the chances of offspring of that particular cross. So they have 100%. Their genotype is 100% big T, little t, and 100% tall. So this is the second Punnett square. Now we're going to look at the trait of shape in peas. There's a round and a big R, and then there's a wrinkled. We're going to use a recessive. So we've got our dominant allele and our recessive allele. We've got letters for them, so we're good, and they're the same letter. So yay, we've done well. Once again, physical characteristic, genetic makeup, genotype and phenotype. If we get a homozygous dominant, big R, that means the phenotype is going to be round. If we get heterozygous, where we've got a dominant with a recessive, we wind up with a phenotype of round. If we get two recessives or homozygous recessive, our phenotype is going to be wrinkled. Okay. Okay, so this in this Punnett square, though, what we're gonna do here is that we're gonna cross a heterozygous round with a homozygous recessive. And the way you set this up, you have to separate the alleles, because the reason why you put them in two different boxes, the big R and the little r, for the first parent, is because when you make eggs or pollen or sperm or whatever it is, gametes, they carry half of your DNA. So that's why we have to separate them into their own boxes, because they basically, it shows that half of the DNA is going into each of the sperm or the egg or the pollen. They're only going to get half of the DNA. So one parent basically puts half of its pollen is going to carry the allele for round, and half of its pollen is going to carry the allele for wrinkled. The other parent, half of its pollen is going to be carrying the allele for wrinkled, and the other half of its pollen is going to be carrying the allele for wrinkled too, because it's homozygous recessive. So that's why we put them in each of those boxes. The next thing you do is that you basically multiply them together. So the first set of boxes, as you can see here, all have, like, the first column of boxes from the result of that cross carry a dominant. So we've got 50% um, heterozygous and 50% homozygous recessive. What phenotypes does this give you? 50% you have a 50% chance of making an offspring that is round. If you do this cross, you have a 50% chance of having offspring that is wrinkled. So if you were to cross these two organisms together, that's what you can bet on. You can bet on that if they have offspring, 50% of them are, that's the chances. 50% of them might be round, 50% of them might be wrinkled. That's what it's telling you. It's giving you the chances that that's gonna happen. So you have a 50, if you cross these, you have a 50-50 chance of getting one or the other. It's like flipping a coin in this case. So the final example here is going to be to do with dimples. I don't think this holds true for humans, but we can go ahead and try it. So once again, the phenotype is the physical characteristics. The genotype is genetic makeup. So the homozygous recessive in this case, the phenotype for that is dimples. The heterozygous basically gives you the phenotype of dimples, and the homozygous recessive gives you the phenotype of no dimples. So that's what we've got right here. So let's go ahead and cross a couple here. Okay, so here we're going to cross heterozygous with dimples with another heterozygous with dimples. So these two individuals have dimples, and but they're carrying a gene that for not having dimples, but they have dimples themselves. So you can see here that if you cross them in the boxes and combine them, you wind up with a phenotype percentage of 25% homozygous dominant, 50% heterozygous, and 25% chances of having a homozygous recessive. The phenotype possibilities are 75% with dimples and 25% with no dimples. So this gives you basically it tells you that if you get two individuals with dimples, but they're heterozygotes each, they have a 75% chance of creating offspring with dimples and a 25% chance of creating offspring with no dimples. So every time they have a baby, they have a 75% chance of having a baby with dimples. They have a 25% chance of having that baby with no dimples. And every single time they have a baby, that's the breakdown of the chances. So for every baby, they're going to have 75% chance that they're going to get dimples and a 25% chance that they're not going to get dimples. That's how it breaks down, and that's how you use the knowledge of this knowledge. Okay, so Punnett squares. What we've been talking about so far is monohybrid. They're called monohybrid crosses because we're looking only at one factor, one trait at a time. And we use Punnett squares for that, and we study two alleles of one gene. So seed color or round or tall. Those are all examples of one trait. So we've been looking at two factors or two alleles and one trait. One factor and two alleles for that one factor. There's also these things called dihybrid crosses. We're going to look at them a little bit closer in a bit. And these are used to cross two factors at a time. So you use these to create giant Punnett squares where you study four alleles at the same time or two genes at a time. So for every gene, there's two alleles. Okay, so you can investigate, for example, seed shape with seed color. So um, organisms or plants that have round seeds that are wrinkled or round seeds that are green uh, and and combine them with yellow wrinkled and yellow green or green and you can find out different combinations. For this, you need the FOIL method or distributive property, and we will be looking into how to do these a little bit later. But if you know how to do monohybrid crosses, that's pretty much what you need to know at this point. And finally, you don't have this slide, but basically here we go. Brown-eyed parents can have blue-eyed child. So here, if you get two parents that are brown-eyed and have heterozygotes, I talked about this before. They do the distributive property or the FOIL method or whichever way you've been taught, basically you could figure out do a Punnett square with them and create this. If the two big B's combine, you get a brown-eyed child. If the big B and the little b combine from big B from one and little b from the other, you get a brown-eyed. If you combine the big B from one parent and the little b from the other little b, you get brown eyes. But if you combine the two little b's, you basically wind up with a blue-eyed. So if these two parents have both brown eyes, this is how they can have a blue-eyed child, but they only have a 25% chance of producing an offspring that has blue eyes. So is it like it doesn't mean they're going to have four kids and one of them's going to have brown eyes. What it means is that for every child that they have a 25% chance of having a blue-eyed child. That's what it means. So that's how you can get some people in a family that have light eyes or light skin or light hair or the combination of all three, because sometimes and nobody else in their family looks like them because for every time everybody in their family that had brown eyes, they had a 25% chance of making a baby with lighter features. They always had the dominant overtaking, and they'd always overshadow the recessive, and only every now and then will they produce offspring that are have lighter eyes, because there's always only a 25% chance of that happening with every child that they have. So it once again, it does not mean they're going to have four children and one of them's going to have blue eyes and three of them are not going to have are not going to have the blue eyes, are going to have brown eyes. It doesn't mean that. It means that for every child, they have a 25% chance of having a blue-eyed child and a 75% chance of having a brown-eyed child. So make sure you understand that, and I hope you were able to keep up with the notes now that they're filling. And make sure you add questions, you add a summary. I've added stuff to the top and the side so you don't have to write everything. Make sure you glue them down to the right pages and then show them to me for a signature so you can get all your points when we correct the notebook. Okay, see you guys next time. Bye.